Hello friends, welcome to ENTRANCEADVISER.com. Today in this video I'm going to talk about the Unit 4, that is the plant physiology, and the most important chapter 13, the photosynthesis in higher plants. Before that, if you are new to this channel, please hit the bell icon and do subscribe to our channel. So let's start. Giving a brief intro of the photosynthesis. The photosynthesis is a process by which the phototropes convert light energy into chemical energy, which is later used to fuel cellular activities. The chemical activity is stored in the form of sugars, which are created from water and carbon dioxide. The photosynthesis is a physico-chemical process by which the green plants use the light energy to drive the synthesis of the organic compounds. It is an enzyme-regulated anabolic process. The photosynthesis is the basis of life on the earth because it is the primary source of all food on the earth, and it is responsible for the release of oxygen in the atmosphere. The chlorophyll, light and the CO2 is required for the photosynthesis. It occurs only in the green part of the leaves and in the presence of light. So all the living organisms need the energy to perform their different normal activities or the normal metabolic functions. Food is the source of energy of the living body. During the oxidation process, respiration, that potential or the stored energy is converted to the kinetic energy, whereby the necessary energy is supplied to the living body. The green chlorophyllous organism produces the food by the means of a special physiological function, and that is the photosynthesis. During this process, the solar energy is stored in the plants or stored within the food as a potential energy. This special physiological function is called the photosynthesis. The photosynthesis or the process is one of the remarkable activities of the living cell of the green plant. So the term photosynthesis was derived from the two terms, photos, which means light, and the synthesis means building up. The green cells synthesize enormous amount of food materials with the help of the light energy, preferably available from the sun. And the synthesis of the food is called the photosynthesis, and the carbohydrates which are produced as a result of the photosynthesis constitute the basic raw materials which directly or indirectly give rise to all types of organic compounds which are needed for the entire living world. So here is the official definition of the photosynthesis, which states that the process exclusively takes place in the chloroplast through the photosynthetic pigments such as chlorophyll A, chlorophyll B, carotene and the xanthophyll. All the green plants and the few other autotrophic organisms utilize the photosynthesis to synthesize nutrients by using the carbon dioxide, water and sunlight. The byproduct of the photosynthesis is oxygen. So during the process of this photosynthesis, light energy is converted into the chemical energy and is stored as the carbohydrates, as you can see in the equation. Here is the equation. You can see 6 CO2 plus 12 H2O, that is after trapping the light energy, 6 C6 H12O6 plus C6 H2O plus 6 O2. Now, why the photosynthesis is called an anabolic process? The metabolic process by means of which increase in dry weight of the body of the living organism occurs is called the anabolic process. In the process of the photosynthesis, the complex substance glucose is produced in the chloroplast due to the utilization of the simpler substances like soil water, atmospheric carbon dioxide, energy of the sun, whereby increase in the dry weight of the plant body takes place. Okay. Hence, the photosynthesis is regarded as the anabolic process. What is carbon assimilation? The inclusion of the carbon from the atmospheric carbon dioxide in the cell component occurs during photosynthesis. So during the dark phase of the photosynthesis, the carbon of the carbon dioxide of the atmosphere is utilized in the preparation of the glucose, that is the C6H12O6, and the carbon assimilation process occurs during the photosynthesis in the presence of light. And hence the photosynthesis is also called the carbon assimilation process. So here are some early experiments or the history, you can say, uh, which are being interesting to learn about those simple experiments which led to the gradual development in our understanding of the photosynthesis. So, in 1845, Julius Robert Mayer was first to recognize that the green plants have the power to utilize the light energy and afterwards convert it into the chemical energy of the organic matter with the evolution of the oxygen. In 1862, Sachs and others stated that starch is the photosynthetic product. So you can see in the 1770, Joseph Priestley performed a series of experiments that revealed the essential role of air in the growth of pattern of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, you can see here in this picture, in a closed space, a bell jar soon gets extinguished, and similarly, a mouse kept a mouse was kept in it and would soon suffocate in the closed space. He concluded that a burning candle or an animal can breathe that air. Both somehow damage the air, but when he placed a mint plant in it continued to burn in the bell jar. And Priestley hypothesized as follows: plants restore to the air whatever breathing animals and burning candles remove. I don't know you get it or not, but I'm explaining once more. And on the basis of his experiments, he showed the essential role of air in the growth of green plants. A mouse kept in a closed space could get suffocated and die, but if a mint plant is kept in the bell jar, neither the candle will extinguish nor will the mouse will die. And he concluded that foul air that was produced by the animal is converted into the pure air by the plants. Priestley discovered oxygen gas in 1774. Next, using a similar setup as the one by the Priestley, but by placing it in once in dark and once in the sunlight, Jan Ingen's house in 1730 to 1799 showed that the sunlight is essential to the plant process that somehow purifies the air fouled by the burning candles or breathing animals. He is an elegant experiment. His experiment was an elegant experiment with an aquatic plant showed that a bright sunlight. Small bubbles were formed around the green plants while in the dark they did not. And later he identified these bubbles to be the oxygen. Hence he showed that it is only the green plants of the plants that can release that oxygen. But later in 1854, Julius von Sachs showed that the green part in the plants produce the glucose, which is stored as a starch. So starch is the visible product of the photosynthesis. As you all know, it was not until about 1854 that Julius von Sachs provided evidence for the production of the glucose when the plants grow. And glucose is usually stored as a starch. His later studies showed that the green substance in the plant, known as chlorophyll, and is located in the spherical bodies, later known as chloroplast, within the plant cells. He found that the green parts in the plants is where the glucose is made, and that the glucose is usually stored as starch. Got it? Then T.W. Engelmann in 1843 to 1909 used a prism to split the light into components and then illuminated the cladophora and algae placed in a suspension of the aerobic bacteria. He found that the bacteria accumulated in the blue and the red light of the split spectrum. He thus discovered the effect of the different wavelength of light on the photosynthesis action spectrum. So, considering the interesting experiments done by T.W. Engelmann, he using a prism, he split the light into its spectral components and then illuminated a green alga, named cladophora, placed in a suspension of aerobic bacteria, and the bacteria were used to detect the sites of the O2 evolution. He observed that the bacteria accumulated mainly in the region of the blue and the red light of the split spectrum, and a first action spectrum of the photosynthesis was thus described then. and it resembles roughly the absorption spectra of the chlorophyll AB. I will discuss it later. And then Cornelius van Niel in 1897 and 1985, on the basis of the studies with the purple and green sulfur bacteria, showed that the photosynthesis is a light-dependent reaction in which the hydrogen forms an oxidizable compound releases to form the sugar. You can see clearly here is a 2H2M plus CO2, light 2A and 6H2O and H2O again. So it was a milestone or to the contribution to the understanding of the photosynthesis that made by a microbiologist Cornelius van Niel in 1897 to 1985, based on the studies of the purple and the green sulfur bacteria, demonstrated that the photosynthesis is essentially a light-dependent reaction in which the hydrogen form a suitable oxidizable compound reduces the carbon dioxide to carbohydrates, and that can be expressed by this reaction only. And by the middle of the 19th century, the key features of the photosynthesis were known, namely the plants could use the light energy to make carbohydrates from carbon dioxide and water. A simple reaction was then known, and the empirical equation, only the CO2 plus H2O light energy falls, and only CH2, only a organic compound formed, plus oxygen. The total process of photosynthesis for oxygen evolving organisms was then understood as just like this, simple, where the CH2 represented a carbohydrate molecule, example of glucose, 6-carbon sugar, etc. Only this. Then in green plants, H2O is the hydrogen donor and is oxidized to O2, the oxygen. Some organisms do not release the oxygen during the photosynthesis. When the hydrogen sulfide or H2S instead of hydrogen donor from a purple or the green sulfur bacteria, the oxidation of the product is the sulfur or the sulfate depending on the organism and not the O2. Hence, he inferred that the O2 evolved by the green plant comes from the H2, not from the carbon dioxide. This was later proved by using radioisotopic techniques, and the correct equation that would represent overall process was the 6CO2 plus 12H2O and 6H12O6, 6H2O and 6O2. As you know from this overall equation, I am coming to this, and the green sulfur bacteria, when H2S instead of H2O was used as a hydrogen donor, no oxygen was evolved. Okay, and he inferred that the O2 evolved by the green plants comes from the H2O but not from the carbon dioxide as thought earlier. I think this is clear to you. You can see the main reaction. The correct equation for the photosynthesis is as follows. I told you earlier. You can see this and memorize it correctly. This is going to come in your NEET and other competitive exams, where the C6H12O6 represents the glucose. The O2 released is from water. That was proved using the radioisotope techniques. And this is not a single reaction but the description of a multi-step process called the photosynthesis. So can you explain why the 12 molecules of the water as the substrate are used in the equation given above? So just think it and write in the comments below. Thanks for watching, and I hope you've enjoyed this video. Stay tuned. Good luck.